Okay, so in our first unit, we talked about chemical bases of life. The smallest unit of matter, atoms, how atoms combine to form molecules, how molecules combine to form big molecules, how big molecules form cells, and how we have two different types of cells, right? Now, in this unit, we learn how those cells make energy to do work. Okay. So first, before we can begin, in today's lecture, we'll talk about energy. Okay, let me get my. Okay. So remember the SLOs for this lecture. When you prepare for your exam, you should be able to answer these questions. Okay? Also, for extra credit for lecture exam 1, some of you just pasted a picture of your index card and I can't read that, I can't give you points on that. It should be legible to me, your answers. Okay. Okay. So first of all, what is energy? Okay. So we can define energy as capacity to do work. Okay. And what does work mean is to bring about movement against an opposing force. Or in short, you can remember work is if you're bringing about any change. Okay. If you are moving an object from this place to this place, that's work, right? Because you're bringing about some kind of change. Now there are two types of energy. Kinetic energy, that means energy in motion, okay, when something is moving. Because you can imagine if something is moving, let's say I'm moving objects, they can cause change, okay, and do work. So I have two examples here. This wrecking ball, when it's moving, it has the energy to break this wall. So this is called kinetic energy. Or you can see this cyclist, as he's cycling, he has the ability to cause change. Okay. Let's say they bump into something. That something will move because there's energy in motion. Okay. So remember, kinetic energy is energy in motion. There's a second type of energy called potential energy. Potential energy means stored energy. So if an object has potential to cause change, it hasn't yet caused change, but if it has the potential to cause change and do work, we call it potential energy. Now it could be due to object's position or structure. So the same example, if we look at this example, we have this wrecking ball, and right now it's not in motion, it's just stationary, so it doesn't have any kinetic energy. But if I take this wrecking ball and lift it up high against gravity and I drop it on a car, will it cause any damage to the car? So it has the potential to do work or cause change, but it's right now at this moment is not in motion. So that's called potential energy, that it's stored energy which has the potential to cause change, but right now it's not causing that change. So for instance, in the last example, we looked at a cyclist in motion with kinetic energy. Now let's say this cyclist is up on a mountain, high up against gravity. So again, this cyclist has the potential to do work when they start biking down gravity, but right now all that energy is just stored energy. Okay, so potential energy converts or changes into kinetic energy when you start moving. So let's consider a few examples to look at what is potential energy, what is kinetic energy. So in each of these examples, how is potential energy stored and how does it change into kinetic energy? So for instance, if you look here, this person is high up here. So, and this person is jumping down gravity. So, which of these people has potential energy? This person, right? The one on the top. And how does it change into kinetic energy? When they jump and they fall in the water, they see a big splash, right? So they have that kinetic energy in motion that will cause change. How about this one? When you stretch this band, does it have potential energy or kinetic energy? And then how does it change into kinetic energy? Yeah, you let it go, it can really actually hurt. The same is true for when you store water up high in a dam. Let's say there's water on the ground. When you bring all that water up high on a tower against gravity, it has a lot of potential energy, right? Now when you let that water fall, it changes into kinetic energy, and actually you can use that kinetic energy even to make electricity, right? If the water falls on a wheel or a turbine, you can move that wheel and then generate electrical energy. Okay? Any questions about potential energy, kinetic energy? Another way you can have potential energy is in sub-objects due to their structures. Okay, so for instance, here you see this structure. Because of this structure, this molecule has potential energy within the bonds of this molecule. So you see this is carbon, this is hydrogen, and this chemical bond, this actually stores energy. So if I were to break this covalent chemical bond, I'll get a release of energy. Okay, and this molecule is actually glucose. Remember, glucose are carbohydrate, and whenever you eat sugar, you get energy out of it, right? So chemical structures can also store energy in their chemical bonds, and this is a kind of potential energy. Okay, and the higher the stored energy, the more complex and unstable the molecule. Okay, so if you look at these three structures, which of them is more complex? This one, or this one? Glucose, right? So this is carbon dioxide, this is water. They're simple molecules, but they can combine together to give you this complex sugar, which is glucose. So as you can see, the structure is more complex. This is unstable, so it stores more energy. Okay. So remember, potential energy, stored energy is due to either position or due to structure. Now in this figure, again this is carbon dioxide, just a gas, water, and this is your glucose, sugar. Which molecule has the most chemical energy? Remember, chemical energy is just stored energy, potential energy. Chemical energy is the energy stored in chemical bonds, like covalent bonds. So which of these three, A, B or C, has the most chemical energy? Okay, 3. And why? Say it again. Most complex structure, okay? It has lots of chemical bonds. If you break them, it releases energy. And actually, what is this process when you're combining carbon dioxide and water and you get sugar? That's what plants do, right? Plants, you have to water them. So they need water. They need air. You can't put a plant inside an airless compartment. So from the air, they take up this gas, which is carbon dioxide. And where does energy come from? How do plants get energy to make sugar?
From the sun.
Sunlight, okay? So they're taking the energy from sunlight and putting all this energy into the chemical bonds to make sugar. And that's why glucose has more energy because all this energy from sunlight is used to make these high energy chemical bonds. Okay, and of course they give out oxygen which we can use to breathe. Okay, so remember this is photosynthesis. Photo is light because we are using light. Synthesis means that you're making something complex. Okay, why do we need energy? Why do cells need energy? Because we have to do a lot of work. Okay, all cells need to do work to be able to survive. Now, what kind of work do cells need to do? Here is an example of mechanical work. Mechanical just means you're moving something. So, for instance, in our muscles, okay, so these are these two different proteins in your muscles. And you can see this is a motor protein, and after you've spent some energy, you'll see this protein moves. Okay, that's how your muscles contract. But the movement of muscles, you need energy. Okay, so that's why we need energy for movement. Another for work that we need energy for is transport. For instance, let's say I was talking about water. Let's say there is water down here and you want to carry this water against gravity up in a high tower. Will you need energy to do that? Let's say you're carrying a bucket of water up a mountain. You need energy to be able to do that. Similarly, in our cells, if you want to carry anything, transport anything against a force, you need energy. So, do you all remember what this is? These little circles and tails? These are the phospholipid membranes, right? These are the hydrophilic heads, the hydrophobic tails, and this is a protein within that plasma membrane. So here you see you have these solutes, these molecules, okay? And you have lots of these molecules here. You have little of this molecule here. So when you want to carry this solute or this molecule from where it is low to where it is high, you need energy. You can't just do it. It's similar to how you're carrying water against gravity. It's very easy to take a bucket of water and just drop it down, right? Because you're going from high, you're going towards gravity. Same here, if you want to carry anything from where it is low to where it is high, you need energy. So if I want to carry this little solute from where it is low to where it is high, I need energy. So that's why your cells need energy to transport things against some force. Here the force is that there is less here and more here. Lastly, you need energy to build complex molecules. Okay. Remember when we talked about macromolecules, we talked about that you have these monomers that combine together to form big polymers. Now, how do you make these big polymers? Because you are making complex structures, you need to input some energy. Okay. So whenever you are synthesizing or building something complex, from simple molecules, you need energy, okay, and that is called synthetic work or chemical work, okay. So, for instance, do you remember the reaction that combines monomers to make polymers? Dehydration, okay. So, here, if I want to take these two molecules and combine them, I need energy, okay. So, for instance, if you want to build proteins from their monomers, which are amino acids, I need energy. It just doesn't happen on its own. Okay. So, we talked about three types of work, right? We talked about mechanical work, which is movement. We talked about transport, and we talked about synthetic or chemical work. So, take a minute. You can talk to partners and try to answer these questions. Which type of work is involved when you're making glucose from simple molecules, and where does this happen in the cell? Remember our organelles. So take a minute, talk to people around you, try to remember where does it happen and what kind of work is it.
I didn't catch what mechanical work is.
Oh, mechanical work? It was contracting muscles. Contracting muscles.
Okay.
Okay? So what type of cellular work is involved? Is it mechanical, transport or synthetic? Synthetic. So synthetic or chemical. And where is this happening? Chloroplast. Remember, only plant cells have chloroplast, and in chloroplast they have these green pigments that can help absorb sunlight and then help combine these simple molecules to make a complex molecule. So this is a plant cell and this is a chloroplast, okay, our organelle. So that's how plants make energy. Plants use it. Okay. Now, there are some chemical reactions that are happening in the cell that are releasing energy, and some chemical reactions that need energy. Okay. So we have specific terms that we use to describe these. The ones that give energy, so for instance, let's say I have glucose, and if I break it down to simple molecules, I'm releasing energy, okay? Such reactions are called exergonic reactions. Now, how do you remember this term? Exer just means out, okay? So what is happening is you're getting some energy out of the reaction, okay? We also call them downhill reactions because they are easy to carry out because you don't need energy. In fact, you're getting energy from them. Okay. So these reactions are called downhill or exergonic. And one example is when you break down complex molecules to simple molecules, you're breaking down bonds and energy is released. Okay. Here is the graph. Okay. And this axis tells you energy. So if you have a bar high up, that means they have more energy. If you have a bar down here, they have less energy. Okay. So remember this terminology. Whenever you have chemical reactions, the starting products are called reactants, and what you get at the end of the reaction are called products. So in exergonic reactions, what happens is your reactants are unstable, they are complex, so they have high energy. And when you break them down, you get products which are low energy. So all this extra energy, the difference between the energy of products and reactants is released. Okay? So you get this output of energy. So for instance, when you break down glucose into carbon dioxide, water, you get energy. Okay? And you can see here, this is the amount of energy released. Now, in contrast, you have endergonic reaction. Ender means inside. So for these reactions, you need an input of energy. You need to provide energy. And we call them uphill reactions. So again, you can think of it as a water example. When you have to lift water up against gravity, it's an endergonic reaction because you're going uphill. But if you just have to drop water downhill, it's exergonic. Okay. So runs uphill because you need energy to build stuff. Okay. So again, this is our graph. This is the potential energy of molecules. Here you can see the reactants are simple molecules. They don't have as much energy. But the products have more energy. They are more complex. So that's why you need energy in this reaction. So you need an input of energy to build these complex molecules from simple ones. And that's why photosynthesis, where plants use sunlight, is an example of an endergonic reaction. Okay. Any questions between exergonic, endergonic? Okay. So if you look at this reaction, you have glucose, which has a complex structure. It has high potential energy. It's being broken down. Is this an endergonic or exergonic reaction? Again, take a minute, talk to people around you before we discuss the answer. Okay, look at all these labels and try to answer this question.
Hopefully that's the right one. If not, I can let you just have this one.
Okay, what did you all decide? Is this an endergonic reaction or an exergonic reaction? Because energy is out. Your glucose has more energy than these products that have low energy. So you get, where does this happen in the cell? Which organelle? Did you discuss that? Which organelle does this happen in? What is the powerhouse of the cell? Mitochondria. Okay. So in plants, animals, everywhere, when they need energy, you can break down glucose to carbon dioxide, water, and you can get energy. And it happens in mitochondria. That's why we call them the powerhouse of energy. And what does this mean, energy out? Where does this energy go? How do we store this energy? Do you remember in our mitochondria lecture, the cellular structures, I said something is the energy currency of the cell. Do you remember that molecule? ATP. So all this energy, you just don't want to let it out, you want to store it. Okay? And then keep it safe and then spend it whenever you need energy. So all this energy is stored in a molecule which we call ATP. Okay, and remember mitochondria is the organelle where all this energy is being made. Okay, so coming to the energy currency of all cells, okay, in bacteria, in archaea, in every domain of life, every cell, the way a cell stores energy is in this molecule called ATP. If ATP stores energy, even before I tell you the structure, do you think it will have a complex structure or a simple structure? Complex, because if it carries all this energy, its structure is complex and unstable. Okay. Before actually we move on to there, do you remember when we talked about DNA, RNA? We said they are made up of these monomers called nucleotides, and nucleotides are made up of 3 components. Do you remember which components are nucleotides made up of? There's a what does this look like? Which macromolecule? It is a sugar, but not glucose. But it's called ribose. But there's a sugar in a nucleotide. Then remember we had a phosphate group. And we had something more that contained nitrogen. Do you remember that? The nitrogen base. So a nitrogen containing base. ATP is not a nucleotide, but its structure is a lot like a nucleotide. That's why I am bringing it back. ATP is a coenzyme, and you've all done the enzyme lab. Again, this is a complicated term, so you can remember it that it's helper. It's an enzyme helper. Okay. So now, if you look at the structure of ATP, this is the sugar. We call it the ribose sugar. Glucose has 6 carbons, ribose has 5. That's why the difference. So that's why it's not a hexagon. You will see it's a pentagon. So it has sugar. It has this nitrogen base, adenine. Okay, remember our A. So these two things are same. The only difference is instead of 1 phosphate group, it has 3 phosphate groups. Okay, so you can see these yellow atoms, they are phosphorus. That's why the name ATP is ATP, because it's adenosine triphosphate. Tri means 3 phosphates. Okay, so that's another way you can remember the structure is there are 3 phosphate groups, and you have this base and this sugar which is called adenosine. So this is called adenosine, and then you have 3 phosphates. Okay, it has a very high potential energy because these bonds that are connecting these phosphate groups, they store a lot of energy. So when you break these bonds, energy is released. Okay, so when you have to make these bonds, you need to give energy. And ATP is the form, it's the currency of the cell that you can then spend to do any work within the cell. Okay, so remember ATP is your energy currency. And ATP undergoes this chemical cycle called ATP and ADP. Again, the only difference between the two is T stands for tri, so 3 phosphates. And when you break one bond to release a phosphate, it becomes di. So it becomes adenosine diphosphate. So if you look here, this is our ATP. So remember, this is our nitrogen base, ribose, and you have 3 phosphates. When you break this last phosphate and release it, see this is released, what happens is you just get a diphosphate. Okay, so this is adenosine triphosphate, this is adenosine diphosphate. Okay, so when you break this molecule, energy is released, and when you have to make this molecule, you need to give energy. Okay, and one of the ways you can remember this is think of ATP as a charged battery in the cell. Okay, so this is ATP is a charged battery. It has 3 phosphate groups. When your cell needs energy, what it can do is it can break it down to make ADP, and ADP is discharged battery. So some of this energy is lost. Okay, so whenever your cells need energy, they'll take ATP, they'll break down this bond, and you can get energy out and use it for transport or whatever. But now you have a discharged battery. So how can you charge this battery back up to make ATP again? How can I charge ADP to form ATP? Remember, you just talked about exergonic reactions. What is happening in mitochondria? You just have to give energy. So for instance, when I'm breaking down glucose into simple molecules, energy is released and you can use that energy to again charge this discharged battery. So the way to remember ATP is charged battery. When you use energy, you discharge the battery, you form this molecule called ADP, and then you have to charge this again using, like breaking down glucose or some high energy molecule. Okay. So remember, ATP is triphosphate, ADP is diphosphate. This is high energy, this is low energy. Another way to think of it is, think of ATP like a compressed spring. Okay, it's unstable, it's compressed, and think of ADP like a relaxed spring. Okay, so this, when you have a compressed spring, it has the ability to do work, it has high energy, and when it does work, it relaxes and all that energy is released. Okay? So, two ways you can remember it is a battery, a rechargeable battery. Okay? Now, cells are continuously spending ATP to do all kinds of work, and you can keep recycling it because when you break ATP, you release energy, you are making ADP, but then you can combine them again using respiration in mitochondria. Okay? Up to 10 million ATPs are consumed each second in a working muscle cell. So we are constantly using ATP and then constantly also making ATP, right? So that's why you need to eat when you're especially working out. Now imagine, let's say I work in a fast food store. Okay, let's say I work in McDonald's. And they say, okay, for all that work, instead of paying you cash or money, all you can do is just buy food at McDonald's. That's not sustainable, right? How will I pay my rent? If all I can do with whatever I'm working there is eat at McDonald's, right? So, that's why ATP, what it can do is, it can transfer its phosphate group to some other molecule, and then that molecule becomes charged and high energy and can do its work. So basically it's like your employer paying you money and then you can use that money for whatever you want to do. Okay? So ATP has this high energy phosphate group and it can then transfer this phosphate group to another molecule, and then that molecule becomes charged and high energy. Okay, so imagine ATP is shaking hand with another molecule and gives all its high energy to that molecule by giving its phosphate group to that molecule. Okay, so for instance, here you will see this is synthetic work, right? Chemical work where you're combining two molecules. And you will see this molecule has a high energy phosphate group. Okay. So when you have to combine these molecules, you need energy. So this high energy phosphate group gives that energy and is then released back. Okay. And you get this combination. Similarly here, do you see this motor protein? What does this motor protein have? This high energy phosphate molecule. So when this muscle protein has to move, all this high energy is transferred to this muscle protein, and then the phosphate, once the energy is transferred, this phosphate group is released. Okay. Same here. can you see this membrane protein has this high energy phosphate molecule? This bond between the phosphate molecule and the protein is high energy. So when you need to transport something uphill, this bond is broken. That energy is used to move this molecule from where it is low to where it is high. Okay. So ATP can be used to, the energy from ATP can be transferred to other molecules by transferring this high energy phosphate group to these molecules, and then they become charged. Does that, I know that's a hard concept, but does that make sense? ATP has this very high energy phosphate group which is bonded to ATP, but ATP can transfer that high energy phosphate to these molecules, and now they have high energy. And when you break this bond, all that high energy is used to do work. Okay? I think I already discussed this, so, oh, no. Where does all this ATP come from? What part of the cell is producing ATP? So we did discuss this. So remember, mitochondria is producing all this ATP, where glucose is broken down into carbon dioxide, water, and all the energy out is used to make ATP. Okay? So remember, when you break the bond of ATP to make ADP, you get energy out, which you use to do work. And then how do you again make ATP? In the mitochondria, you do something called respiration, where you're breaking down glucose and all the energy is used to then again form ATP. So remember this cycle. This is our charged battery. It discharges, but then we can charge it back again. Okay? Now, how is energy used by living things? In our body, as I said, in our cell, there's so many chemical reactions going on. Some of them release energy, some of them use energy. So what our cell does, it combines these reactions. So let's say I have a reaction which has an energy output. So I can use the energy, this energy that has been released, to do a reaction that needs energy. So these reactions we say are coupled. Okay? Let's say, I have a molecule. This is a reactant. When it converts into B, you get energy out. So if I have another reaction where this molecule, it needs energy to form D, I can use this energy to drive this reaction. So this is called coupling. That you're coupling an exergonic reaction which releases energy to an endergonic reaction which needs energy. So that's how our cell couples these reactions. So energy in, so when exergonic reactions, they fund endergonic reactions. So here is an exergonic reaction. This is energy. What is happening is you're taking high energy molecules, breaking them down to low energy molecules. So your energy is released. And you can use this energy to then do a reaction where you need energy to make these products from these low energy reactants. Okay? So you can combine them. It's like, let's say I'm working for an employer, I get paid. So that's energy out. Now I have money, but I can invest that money to maybe start a business which needs money. Okay? So for instance, here, combining two molecules to form a complex molecule, that's an endergonic reaction. It needs energy. And how do you get that energy? By breaking this high energy phosphate bond. Again, here, I need energy to move this muscle protein for contraction. So that's an endergonic reaction. How am I getting energy? By breaking this high energy phosphate bond, the energy is released and this muscle can be moved. Same here, moving these molecules from low to high, it's an endergonic reaction. I need energy. How do I get that energy? By breaking this high energy phosphate bond, energy is released and can be used. Okay. Take a minute to look through your notes before we move on to enzymes. Is anything unclear or does it make sense, or any word that I used that was just out there and didn't make sense? What energy do I have now?
Kinetic.
Kinetic. If I stand up here, what energy do I have? I'm not going to stand up here. Potential. Okay. Moving on to enzymes. No questions? So this is something, hopefully because of last week's lab, might be a little easier to understand because you did work with an actual enzyme, you had starting products, you starting molecules, end products, and you saw what happens, right? You worked at the enzyme at different pH, at different temperature. So hopefully this will not be too new. So this is just a graphic of all the chemical reactions that are taking place in a cell. Remember, chemical reactions are nothing. You have reactants, your starting products, and then you have end products, what you end with. So many of these reactions are happening in your cell. Bonds are being broken, bonds are being made, and there's so many. And all these reactions together, we call it metabolism. So metabolism is the sum total of all chemical reactions that are taking place in a cell. Okay? And all these reactions are not spontaneously happening in the cell. Okay? They need help from enzymes. Okay? So our cells, each cell, they have thousands of different enzymes that are helping each of these chemical reactions. Okay, so that's why it's pretty important to learn what enzymes are, because without them, the cell will come to a standstill. It won't be able to carry out these reactions. You won't be able to make energy. If you can't make energy, you won't be able to do any work. So that's why enzymes are important. Usually all enzymes are proteins. Remember, we talked about macromolecules. One of the category is proteins. They have lots of these different functions, and enzymes is one of them. So enzymes are proteins. Okay. Now, actually, this was the question I asked you on exam. Maybe I'll ask it again on exam too, because I want you to remember this. Remember the 4 levels of protein structure. What is the first level? Primary. And what are these little blue boxes? What are the monomers? Amino acids. Okay. So these amino acids, they combine together by covalent bonds. Do you remember what the covalent bond is called?
Peptide bond.
Peptide bond. Okay. So these amino acids, they combine by peptide bonds to pretty much just form a chain, right? It's a linear chain. Remember our beads of a necklace? These are just little beads on a necklace. It's just a straight polymer. Okay. So this is the first stage. Then what happens is it starts forming these. Actually, before I can move on, if you remember the structure of an amino acid, carbon can form 4 covalent bonds. One bond is hydrogen. Another is this amino group, carboxyl group. And even if you forget all this, remember what makes each amino acid different is this R group. So how many amino acids do we have? 20. Okay, just like remember we have 26 alphabets in English, we have 20 amino acids. Each amino acid has this different, here you can have different molecules, different chemicals that makes them different. So when we have all these amino acids linked together, these chemicals, they start twisting and forming bonds. And that's why they start forming these structures. Like here you see this helix or these sheets. Okay. And if you look carefully here, can you read what kind of bonds are making these structures? Hydrogen bonds. So between these different R groups, you start forming hydrogen bonds, and that's why you get these helices, these pleats. Remember one of the questions on exam was, can you name a macromolecule with hydrogen bonds? So the answer could have been nucleic acids, proteins. Then this is the second level or the secondary structure. Then you have a tertiary structure where you then form more of these bonds and you get like a ball-like 3D structure. And sometimes in some proteins, multiple balls come together to form the quaternary structure. So that's what enzymes do as well. They have primary, secondary, and tertiary structure. Sometimes they'll have quaternary structure, not all of them. So once they form this 3D structure, this is how an enzyme looks. And this is, remember, the pocket or the active site where your reactant or your substrate can go in. So enzymes are nothing but proteins. I guess we already discussed this. What type of bond holds amino acids together? What is this bond? Peptide bond, right? This is covalent bond, peptide bond. See, this is one amino acid and you can see these 3 groups are the same. Amino group, carboxyl group, hydrogen. What is different is this green block. That's your R group. And these 2 amino acids can form a covalent bond and we call this peptide bond. And just like that, you get the first level where we have these different amino acids are all strung together in a linear polymer. Okay. And remember, this is primary structure, but then once it folds, it becomes a tertiary structure. Now again, remember, enzymes are proteins and they are actually catalysts. What does the name catalyst means is they help speed up a reaction. So they can help start a reaction and speed it up. So we say they initiate reactions and they accelerate reactions. So here you can see you need enzymes for all kinds of reactions. Here you see these are your starting molecule or substrate. This is your product. So your substrates here, they need to be combined together to form a product. So an enzyme can do that, make complex molecules, and enzymes can also break complex molecules into simple ones. So the enzyme you studied in lab, what kind of enzyme was it? Was it breaking down a complex molecule or making a complex molecule? Do you remember what the name of the enzyme was? Lactase. This could be on your exam today. And then it was breaking down the disaccharide into galactose and glucose. Remember, in our lab we used a different substrate, but So that's what enzymes do. Any kind of reaction, they help start the reaction and speed it up. Okay. Usually you will see all enzymes, they end with this suffix called ase. So lactase, lipase, nuclease. And the first half of the enzyme is usually the substrate. So for instance, lactase is an enzyme that works on the substrate lactose. Lipase is an enzyme that works on the on lipids. Nuclease is an enzyme that works on nucleic acid. DNA polymerase is an enzyme that builds DNA, makes the DNA polymer. Okay? So the name always tells you what the function is. So what do you think the function of an ATP synthase is? Synthesize ATP. So it makes ATP from ADP and the phosphate. What do you think the function of a carboxylase is? I know that's a hard one. A carboxylase actually adds carbon dioxide to a molecule. Okay? Okay? Again, this is something we saw in our lab. This is an enzyme. Remember, it has this pocket where the substrate fits in. Okay? Now, the substrate here is broken down. You get products that are released. So, all enzymes have this pocket which is very, very specific for the substrate. Not just anything can bind in. Each enzyme is very specific to which substrate it could bind in, and in the first step they form this complex which is called enzyme substrate complex. Okay. Again, try to answer this. Enzymes are highly specific and can only bind their own specific substrate. Is that true or is that false? So remember, that is true. This little pocket can only bind a specific substrate. Okay. Now here again you will see this is an enzyme. This is your substrate, and you will see the substrate goes into the pocket and the enzyme sort of folds around it to form a snug fit. So for instance, if you, in a lab, if you've ever worn gloves, or not just in lab, whenever you wear gloves just come in like a free size, right? But when you put it on your hand, it sort of snugly just fits in your hand and takes the shape of your hand. Just like that, in enzymes, when the substrate comes in, it sort of folds around that substrate. Okay? And we call that induced fit, because as the substrate is coming in, the enzyme sort of forms an induced fit around the substrate. Okay? So here you can see that the enzyme sort of fits around the substrate. So the entry of the substrate induces the enzyme to change shape slightly, making the fit between the substrate and the active site snugger. So you can remember it like a glove fitting a hand. Okay? It's not very rigid that, oh, there's a pocket side, substrate fits in. It's more dynamic, where the substrate comes in and the enzyme sort of forms a snug fit. So let's go through our steps. This is our enzyme. Again, it's lactase. This is the active site and it's empty. It's ready for the substrate. Now, this is lactose, our disaccharide. These two monosaccharides are attached by this bond. Now, this substrate, it fits in. Remember, this is enzyme substrate complex. Now you have catalysis. That means the reaction is being sped up. Remember, when you're breaking these bonds, this is called hydrolysis. You add in water molecule, you break this bond, you get products. These products are released outside And in the end, the most important thing is now the enzyme is free and empty again. So an enzyme can keep on doing this. Okay. So remember, they can be used again and again because they are not altered. You get back the original enzyme at the end of the reaction. This is something, again, it's very, very important even for your last week's lab and for this class. So again, take a minute or two to discuss with people around you. Would an enzyme work if it were a polypeptide? First of all, what does a polypeptide mean? This first stage where your protein hasn't folded, that's a polypeptide. Once it's folded, it's called a protein. Okay. Why or why not? Do you think an enzyme will work if you just have a linear string? So take a minute, discuss it because this is an important question.
It won't work because in a linear structure, the protein isn't functional. And so they wouldn't be able to attach with the substrate. What about you?
But that sounds like a good explanation.
So what is the answer? Do you think it will work as a polypeptide? What is the consensus? Do you think an enzyme will form an active site if it's just a straight line? It won't work if it's just this first level of structure. There is no pocket, no active site. So the chemistry won't take place. Your substrate can't fit in. And you can see in this GIF, this is just a polypeptide, but it has to fold into the tertiary three-dimensional structure to be functional. Otherwise, an enzyme is not functional. Okay. So that's why conditions that, so let's say I have an enzyme and if I change some conditions that makes it go back to this structure, it won't be able to function. And that's what you did in the lab. When you changed temperature and you changed pH. So for instance, here is this graph and you can look at temperature. So this is lowest temperature and you're increasing the temperature as you go towards right. And here is how fast the reaction is. So rate of reaction means how fast or how quickly the product is forming. So if you look at your enzyme, you see that at this temperature it works the best because you get the maximum product. But if you go either way, if you lower the temperature or if you make the temperature high, the enzyme is not working at its best. Okay, and we saw that in our lab too, that in ice you don't get any activity, and when you boil the enzyme you don't get activity. There's always this optimum best range where the enzyme works because at this range your enzyme has the tertiary structure. So two of these factors that govern enzyme action are temperature, pH. Okay. So all enzymes have these optimal ranges. What does optimal means? That where the enzyme works the best. Okay. Where they operate the best, and outside of that physiological range, the enzymes can stop working. Okay. So here we looked at temperature. This is pH for two different enzymes, pepsin and trypsin. So again, this is pH. So this is lowest, this is most acidic, and this is highest, this is most basic. So you can see here, pepsin is an enzyme that works in acidic pH. So within your body, where do you think pepsin is? In the stomach. Because your stomach is acidic and pepsin is fine. It maintains its structure and is able to catalyze reaction within stomach where there is acidic pH. Whereas this other enzyme, trypsin, it works near neutral pH. Neutral is 7, so it works around neutral pH. So it will not be able to function in this in the stomach. So each enzyme actually has its own optimal range. It's not constant for every enzyme. It's different for each enzyme. Okay. And if you raise the temperature or lower the temperature, raise or lower pH, beyond this physiological limit, what it does it denatures the enzyme, meaning that your 3D structure is destroyed and your protein goes back to the linear polypeptide. It still has all the covalent bonds, but it's unfolded. Okay, it doesn't have the 3D structure that it forms with all those hydrogen bonds. Okay. So what happens at high temperatures? Remember, let's say all these are little molecules. When you increase temperature or heat something, these molecules start moving more. And when these molecules start moving more, you end up disrupting these chemical bonds within protein. So what you're doing is your folded protein is getting denatured and you form this unfolded protein. So the 3D shape is broken. So that's why when you boil the enzyme, it's not functioning. Okay? What happens at cold temperatures is you're slowing down the enzyme so much that the enzyme is not able to even move and get to the substrate and help the reaction. So cold temperatures, they slow down chemical reactions to the point where the enzymes are not even able to function. Okay. Again, we already discussed this. These are two different enzymes with two different optimal pH. One is acidic, one is near neutral. And why is that? Because this is your stomach where pepsin is found, where your stomach is acidic, whereas trypsin is synthesized or made in your pancreas where the pH is near neutral. Okay. Again, what happens when you change the pH? So you can see this is your linear polypeptide. And the way this polypeptide folds is you can see it forms all these bonds. So this is an ionic bond, this is a hydrogen bond. So using all these bonds, the peptide folds. So when you change the pH, you disrupt all these chemical bonds and then the polypeptide becomes unfolded, denatured and is not able to catalyze the reaction. Okay. Does that make sense? So temperature, pH, basically what they are doing is breaking all these bonds in the tertiary 3D structure and unfolding the protein so that now the enzyme cannot function. Okay, so now how do enzymes help reactions? What is the strategy they use to help these different kinds of reactions? Now, any kind of reaction needs a little bit of energy, okay? So, let's say these are my reactants, these are my products, and there is an energy difference. You can see it's an exergonic reaction, right? Reactants have more energy, products have less energy. So you will get an output of energy, but still, let's say let's say you spilled some gas, okay? You go to the gas station, you spill some gas. Is there a fire there immediately?
No.
Unless you actually have a light, there won't be a fire, right? That's the same case with a chemical reaction. Even if overall this is an exergonic reaction, I should get energy output. It's not like spontaneously things will start happening. It's not like spontaneously, things will start burning if there is gas spill. So this little energy that you need to start a reaction, we call it activation energy. Okay. So this is the energy required to start or initiate a chemical reaction. So how enzymes help is
if exergonic reactions occur spontaneously, what keeps molecules from breaking apart and cell chemistry from racing out of control? For any reaction to occur, even a downhill reaction, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, E sub A. This required energy input represents a barrier that prevents even energy-releasing exergonic reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules, referred to as the enzyme substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction.
Okay, so what the enzyme does is this activation energy, when the substrates bind to the enzyme, this is lower. So you need less activation energy to carry out this reaction. So without an enzyme, let's say this is the activation energy you need. But when the reactants bind to a substrate, this is lower. So they lower the activation energy needed to start this reaction. Okay. So again here, if you look at these graphs together, let's say these are reactants, product, that's the difference of energy. Without the enzyme, you need this activation energy, but with, you have lowered it down here. So that's how enzymes help reaction occur quickly. And how are they lowering this activation energy? Because the reactants are getting concentrated, they are brought together into this pocket site. So then it's easy to carry out the reaction. Okay. Let's say I have to combine two things to form one thing. What enzyme is doing is bringing these two things together, so now it's easy to combine them. Okay. They are oriented, so let's say I have to combine this side of the molecule with this side, so the enzymes help them orient in the right direction so that things are not just flowing everywhere. And they also strain bonds. Let's say in the case of lactose, when you have to break the bond, the way the enzyme holds the molecule, the bond is now a little strained, and that's why it's easier to break down. So after the products are released from the active site, the enzyme can accept another molecule, and remember the enzyme can keep on functioning and does not change in a reaction. So factors that influence the amount of product an enzyme turns out: pH, remember it disrupts the bonding in the 3D structure; temperature, there's too much kinetic movement which can again disrupt the bonding. Another thing that affects enzymes are these genetic mutations. Sometimes if there are changes in the DNA, remember DNA is the one that has information to make protein. So if you change the information in DNA, your protein might be different, and if your protein is different, sometimes it doesn't fold properly to form a functional enzyme. Oh, and lastly, the concentration of substrate and enzyme. The more enzyme you have, the more you can catalyze the reaction, or the more substrate you have, the more you can catalyze the reaction. So just try and answer this question and then we'll stop for today. Which of the following scenarios would most likely cause a protein to denature? 1, 2 or 3. Body temperature rises by 1 degree Celsius. Moving the cell from a neutral solution to acidic. A mutation occurs but it does not change the folding of a protein by 1 degree Celsius. So not A, because just 1 degree Celsius, remember when we looked at the graph where enzyme functions, just a couple of degrees doesn't make a difference. But yes, if you go from a pH of 7 to acidic, your protein will unfold. And sometimes you change the DNA, but if the folding doesn't change, your enzyme is still functioning.